Our group did a presentation on a journal titled Gastrointestinal Helminth Parasites of Pet and Stray Dogs as a Potential Risk for Human Health in Bahir Dar Town, northwestern Ethiopia. I'm Sarah Wallace. The other people in my group are Caleb Prater, Matt Palter, and Jake Hayes. So to start off, I'm just going to give you a little bit of background on gastrointestinal parasites. Gastrointestinal helminth parasites of pet and stray dogs causes potential risk for human health. Dogs have been at the right hand of man since cave dwelling times, which makes it very easy for them to carry and spread zoonotic diseases. If you think about it, when you have a dog beside you, it's licking you, you're touching it, you kind of lose track of how much interaction you're actually having with that animal, which causes this increase in potential for risk in infection. In society, dogs are used for companionship, hunting purposes, and commercial purposes, and all of these environments that they're involved in exposes them to infection, to internal parasites. Individuals get infected due to fecal contamination. And when you ask yourself, how does this happen, it's not that you're like eating poop, that's what I think of, but it's just having, for example, when you work out at the barn here at the University of Findlay, when you're working with pigs, yeah, you wash your hands, but there's still a little bit under your fingernails, and when you take your shoes off, you're contaminating yourself again. Then when you go to eat food, you contaminate yourself, and they can take residence in your gut. We know that this contamination is very common in developing countries such as Ethiopia, and that is where a majority of infected individuals live. In these countries, they have more stray dogs, they have more contaminated areas like poor sewage and poor quality of water, and these serve as soil habitats for these parasites, which have a long incubation period and can live in soil without a host for a long time. Once they come into contact with humans, they reside in the digestive tract. There are multiple forms of these internal parasites, otherwise known as helminths. A helminth is a many-celled worm, which can be small and even microscopic, which causes problems once established in the digestive tract. A few examples of these helminths are nematodes or roundworms, cestodes, tapeworms, trematodes, flatworms, and like I said, they reside in the gut. Once established in the gut, these parasites cause side effects similar to crypto, which, as I mentioned before, is very common out of the university's barn if you come into contact with fecal matter. Side effects include diarrhea, a lowered resistance to infection, stunted growth, and other problems, which can lead up to amoebosis, which is an infection caused by an amoeba, a gastrointestinal infection that may or may not be symptomatic and can remain latent in an infected person for several years. So sometimes you don't even know when you're infected, and these internal parasites can get into your bloodstream and spread further, like to your liver, and cause further problems and complications. Amoebosis is the third leading cause of death from parasites worldwide, and the World Health Organization approximates that 50 million people suffer worldwide yearly, and 40 to 100,000 deaths occur annually because of this disease. When that quantity of individuals is affected in an area, it causes the economic growth in that area to be hindered. And that's what we're seeing in Bahir Dar, Ethiopia.
In this paper, a cross-sectional study was done. A cross-sectional study is a form of a class of research methods that involve observation of a population or a representative subset at one specific point in time. It's descriptive, it's not experimental, and it's used to describe prevalence on many occasions. This particular study was done from November 2011 to April 2012 in Bahir Dar Town, northwestern Ethiopia, which is bordered by Tana and the Blue Nile. As you can see in this picture here, you can see the Blue Nile and the Tana, which is the largest lake and largest river in Ethiopia. And also you can see to the east of Somalia and to the west of Sudan. As for the study animals, there were Bahir Dar native dogs, which approximately 1,670 pet dogs and 100 stray dogs. For the experiment, dogs were randomly selected for the sample, and it included dogs of all age groups and sexes. The sample size was determined according to thrust field, given in a formula in his book, Veterinary Epidemiology. He gives several formulas that can be used, and in this particular article, the author simply used the simple sampling formula. Because there was no information on the prevalence of the parasites, a prediction of 50% expected prevalence with 5% absolute precision was made. Approximately 384 pet dogs and 46 stray dogs were randomly selected. As for the sample collection, fecal samples were collected from the rectums of the 384 pet dogs and 46 stray dogs. They were stored in a refrigerator and processed within 2 to 3 hours of collection at the Bahir Dar Regional Animal Disease Diagnosis and Investigation Center. Used was the direct smear, sedimentation, salt flotation technique, and identification of the parasite eggs was used according to the morphological characteristics and key by Soulsby. The first was direct smear. It's done by collecting a small amount of feces on a slide using a type of liquid which depends on how you want to accomplish the technique. If you're looking for helminth eggs, as done in this research project, you'd use water or iodine. You would cover it with a cover slip, move the cover slip around until it lays flat, and you should be able to read through the smear underneath a microscope. Next is sedimentation. It's a treatment process in which the velocity of the water is lowered below the suspension velocity, and the suspended particles settle out of the water due to gravity. The process is known as settling or clarification. The next technique was the salt flotation technique. A small amount of feces is mixed with about 10 milliliters of flotation medium and poured into a tube so that the liquid comes just over the top. The mixture is allowed to sit for 15 minutes while the eggs float to the top and rest as the fecal matter sinks to the bottom. A cover slip can then be placed on the top of the tube before the incubation period starts, or can be applied at the end. The cover slip is then transferred to a microscopic slide and viewed under a microscope. Commercial devices have a sieve incorporated into the tube to keep large particles from floating up and sticking to the cover slip. As for the analysis, all the raw material was entered into Microsoft Excel using descriptive statistics. The calculated prevalence was calculated by the number of infected divided by the total number of individuals, multiplied by 100%. The chi-square formula assessed the risk factors of the parasitic prevalence. SPSS statistical software with 95% confidence level to determine the significant difference was used, and only the p-value is recorded for simplicity. And now I'll have Matt report some of the results.
Okay, to start out, we'll say that the results are classified into certain categories. They tested different breeds of dogs, sex of dogs, male and female, and then they did 3 different age groups, which I'll explain in a little bit. The first, overall, the prevalence was not significant because the pet dog had about a 75% prevalence, and the stray dogs had about an 84%. Like I said, there's no significant difference between those two because there's such a small percentage between them. Then a lot of them had 1 or more infections, which are the concurrent infection is about 73%, so there's more than half had the 2 or more parasites. And then the highest prevalence between both the stray and the pet dogs was the Anaclystoma carninium parasite. And then these are the tables that was given in the results section of the article. As you can see, they have the pet and the stray dogs, how many were examined, how many were positive, and the prevalence for each of them. And then they list the top 7 parasites between the two. You can see that the caninium was the most common between the two, and then the Toxica canis was the next one down. And then this one lists the other. This one gives the other categories that they classified the results in. First, they did the sex of the male and the female. Those prevalences were approximately the 74 for the males and the 76 for the females. Again, since they are so close, they didn't count that as a large enough prevalence to count them significantly different. Then they went on to the age, which they classified the ages into 3 categories: the pups, the younglings, and then the adults. They said the pups were from less than 6 months of age old. The young ones were between 6 months and 1 year old, and then the adults were older than 1 year of age. They had the prevalence among the different age groups weren't that significantly different, but if you do see that they did have the younger ones that were infected were higher because the physiology of the animal, because they don't have the immune response to the parasite that they get from their parents, and it hasn't built up in the body yet, which they would get with age. Next, they did like the breeds of dogs, or like they did a local species, a cross species, and then the exotic breed. Those numbers were the local was a 76% significance, the exotic was a 73, and the crossbred was a 76. These are like a summary of all the stats that the charts gave. And then each of them doesn't have a significant difference. But yeah, when they get to the age, they did say there was a significance because of the young ones compared to the adults. Like the younger they are, the higher the percentage of infected. Then they do go into talking about the feeding systems, which is important because if we don't control their feeding, we don't know what's contaminated and what's not. So they said a controlled feeding was lower than an uncontrolled feeding, which the controlled is like we monitored their feeding and we know what they're eating. And uncontrolled is they just scavenge around in the scraps for the dumpster and whatnot. So obviously the uncontrolled would be higher because those are more contaminated foods and unsanitized. And then just like bringing all this together and discuss what it means. The differences in the parasite prevalences. One is the climate factors. The 2 is the biology of the parasite itself. 2, the facilities that the animals are housed in this case the veterinary facilities. And then the public's view and awareness of the care of the dogs and the animals. And then, like I said before, the physiology of the animal. The health, the hygiene is the #1 issue with controlling the parasite population and infection. The place where the experiment was taking place, they had noticed that the stray dogs were scavenging around like butcher shops and in alleys and like the thrown away animal flesh that the butchers have been throwing on the streets. So that would be the uncontrolled feeding. And then, of course, you're going to have parasites and like such as maggots or something like that that grow in the carcass that will transfer to the pups and the adults. And then we also need to control the feeding. The physiology of the animal. The age, like I said, the young pups don't have their immunosystem up to par yet because they're still growing. Also, if they have a single to repeated exposure of the parasite, if they're exposed to it more, they're going to be more acceptable to it and be infected by that parasite. And then also during birth and right after birth, you have the placental transfer and then the mammary transfer through the milk of the animal, in which they get the immunoglobulins that come with the colostrum. All those help the immune system grow as they get older, so it has to build up in their system. And then I'll have Jake conclude the article.
Awesome. So now that we've heard about what the study was about and all the things they looked at for the study, let's take a moment to figure out what all this actually means. Just to recap, the study that was being done was on the gastrointestinal helminth parasite, which was found to be highly prevalent in both stray and pet dogs in Ethiopia. The biggest concern with this parasite is that it can become zoonotic, which means it can be spread from animals to people, and that causes a lot of issues. A few facts about the disease, just to recap. Over 50 million people are affected by this each year. It is a parasite. It takes hold in the intestinal tract. Importantly, there is no statistical difference between number of pets and the number of stray dogs that are infected with the parasite, which goes to show that just because a dog is in a home doesn't give it any advantage over a dog in the streets. And Ancylostoma canium is the most common form of the parasite. Also found in the study that there's no difference between infection rates in the different sexes of dogs, so males and females are equally as infected. Also, there's no difference between the different breeds of dogs that are affected. Studies did show that more dogs were affected in uncontrolled feeding situations compared to controlled feeding situations. So when we get to prevention, one of the big ways to prevent it would be to control the pet's food, not to let them scavenge in the streets, to make sure they have monitored feeding times. Also, you want to do regular fecal sampling and screening to make sure they're not infected with the parasite or the worms. And along with that, you want regular and strategic deworming practices. Those are usually given to you by veterinarians, and those just include usually oral medications or topical medications that kill the worms in different stages of their life cycle to keep them from coming back and infecting the same dog again and spreading it to other dogs as well. And the most important thing is to increase the pet owner education. The more pet owners know about this issue, the better they're going to be at preventing it. A lot of people don't realize that their dogs are infected with worms, and then that can get spread to the people because they just don't know. And it's not that they're doing anything wrong or meaning to do it wrong, they just aren't aware of what's going on. So increasing the pet owner's education and understanding of the topic is going to be a big help in preventing the spread of this parasite. Another big one would be controlling the animal population. Spaying and neutering your pets. The more strays they have in the streets, the less are in your home. So if there's more in the street, they're not going to have the attention that could possibly be given to them if they were in a home. The stray population is fed by overpopulating, so by spaying and neutering your pets, you're reducing the number of animals that are roaming the streets, and therefore reducing the number of animals that can potentially be contaminated with the parasite and or spread it to other animals. And then the big thing is to treat the stray dogs as well as the pets, because if you treat just the pets, then the strays are still out there, and sooner or later a pet is going to come in contact with a stray, and that can just get the cycle started again, or a person is going to come in contact with a stray and going to get the parasite transmitted to them. Excuse me. You want to fight the spread of zoonotic illness by making sure you wash your hands. The biggest way it's spread, especially with internal parasites, is through fecal matter. So you want to make sure you don't have fecal contamination on your hands. Like Sarah said, you don't want to poop under your nails. So make sure you wash your hands really well before touching animals, after touching animals, even after you take off your clothes that you were wearing when you were around them. And just the big thing is not allowing that fecal contamination to get into your mouth. So if it gets in your mouth, that's how the eggs get inside and that's how they take hold. So that's what you want to prevent. And you also want to treat all your pets. So even if your dog's infected, you also want to treat your cat or your guinea pig or whatever, because it can be transmitted to other animals other than dogs. And another prevention is to treat them at an early age. As Matt said, most of the animals that are affected, they're more prone to be infected with the parasite at a young age. So if you get them started early, then you run less of a risk of them containing the disease and passing it on to people. And, good Lord, human hygiene helps prevent the spread of zoonotic diseases. So if you're cleaning up after yourself, then you're not going to get it. Because it is zoonotic, you can actually spread it to your dog. So if you're clean and your dog's clean, then you have less of a chance of getting it. And like I said before, the most important thing is to educate, so you know what's going on and how to treat it. And that's the end, so thank you.